I know we've talked about this. You did this when you did the letter E. You made a wet mount, but just going to add a little bit more here. So how do you make a wet mount slide? So first off, a wet mount is used when you have some sort of specimen and you want to put it on a slide and view it under the compound microscope. Okay? So to do it, the steps are pretty simple. You start off with just your blank slide, your empty glass slide. And then you start with either a drop or two of water or a drop or two of stain. And we'll talk about that in a second. You put that on the slide. Because for a lot of cell organelles, they don't have a color. They're sort of clear, transparent. And so if you want to see certain organelles, it's difficult to see. So sometimes we use what we call a stain. And there's lots of different types of stains. Today we'll be using iodine and methylene blue as two different stains. But a stain is basically just a chemical substance that colors certain organelles or certain parts so that you can see them easier. So for example, this right here, this is methylene blue, okay, and it's a blue stain, and it will stain the cytoplasm of a cell, sort of a light blue. It will stain the nucleus of the cell a darker blue. So when you add this to a cell, it just makes it a lot easier to actually see the cells and see some of the parts. So you put a drop of stain or a drop of water on your slide. You get a thin slice or a thin sample of whatever it is you want to see. Remember, for the compound microscope, it has to be thin enough for light to pass through it. You put your specimen in the water or in the stain. What's the last step? Jared? Yeah, you cover it with a cover slip. Okay? Remember, when you do put the cover slip, put it on at an angle to force the air out so you don't trap air bubbles under there. And then you'll have your slide made. Look at it on the compound microscope and hopefully see things clearly. So that's all. Those are the steps. Put your liquid, either stain or water, add your specimen, cover with a cover slip, take it to your microscope and look at it. And like I said, today we're going to be doing 3 different times, today and tomorrow. We're going to be viewing 2 types of plant cells and 1 type of animal cell.